At first glance, the left and right sides of the brain appear symmetrical, but in fact, most brains are not. There are tiny differences in the structure and function between the two sides. Do you want to experience this asymmetry for yourself? Alright, straighten your arm in front of you and fixate both eyes on your thumb like this. Then, close one eye at a time, alternating between the two. One eye will probably see a shifted scene. This phenomenon is called ocular dominance and results from asymmetric processing of visual information in your brain. Brain asymmetry also occurs in other animals, such as chicks. The right eye is tuned for discriminating food, while the left eye is tuned for detecting predators. So, where in the brain does asymmetry emerge? We tried to answer this question by studying whether odour information is encoded symmetrically or asymmetrically in the honeybee brain. Bees can be trained to extend their mouth parts to an odour by pairing this odour with a sugar reward. This is called PER conditioning, proboscis extension reflex conditioning. Honeybees detect odours with their antennae. From the antenna, olfactory receptor neurons project to subcompartments of the ipsilateral antennal lobe, called glomeruli. We focus on a specific population of neurons within these glomeruli, the projection neurons. These are output neurons that convey odour information from the glomeruli to higher brain areas. We analysed data obtained at the University of Constance. Under the microscope, bees' brains were imaged and the activity of the projection neurons recorded when an odour was presented. We found that the glomerular activity pattern distances between odours were higher in the right than in the left AL of the brain. So, what were the glomerular activity pattern distances? The glomerular activity pattern distances that we calculated have as many dimensions as number of recorded glomeruli. To understand what odour distances mean, imagine that there would only be two glomeruli. The intensity of the signals invoked by two odours differed less in the left antennal lobe than in the right antennal lobe. So, what does this inter-odour distance represent for the bee? These odour distances correlate well with the capacity of the bee to discriminate two odours during the PER paradigm. We therefore tested the hypothesis that bees can discriminate two odours better than when smelling with the right side of the brain. Bees with only one antenna in use were put in front of an airstream with an odour as background for a few seconds, and were trained to extend their mouth parts when a different target odour was added. So, only the bees that could perceive the target odour as being different could learn to respond to target odour. As hypothesized from the imaging data, bees with the right antenna perform better compared to bees with their left antenna in use. So, why should a bee smell differently with its left and right antenna? One hypothesis is that this asymmetry in odour processing supports two important but opposing functions at a time, which is generalization and discrimination among different odours. Thus, asymmetric coding of symmetric stimuli might allow parallel processing of different stimulus features at the same time.